Hi, this is Professor Schur. This video is going to cover the basics of lipids for our class. It's going to talk about the three types that are most important biologically, what their structures are, what their functions are, and how to identify each of them. So if we start talking about lipids as a whole, as a group, it's actually not a polymer group. So our other large macromolecules are polymer groups, but lipids are not. And you'll see that it's because lipids don't have repeating subunits that are covalently bonded. Of course, they have parts that are bonded together, but it's more diverse than the other molecules as far as shape goes, and it's not repeating of similar or identical building blocks. So why do we put them all together? So the reason we put them together is because they are all hydrophobic to a large extent. Sometimes they might be a little hydrophobic, a little hydrophilic, but the hydrophobicity, the not hydrogen bonding to water, that's why they're all together. So what you're going to look for when you're trying to decide if something's a lipid is you look for your carbons and hydrogens and just a few oxygens. So it's the same elements in large part that make up carbohydrates. However, carbohydrates have many more oxygens and hydroxyl groups. Really what we're looking for is lots of carbon, hydrogen, nonpolar bonds, and that's going to tell you it's a lipid. We have three main categories we're going to discuss. There are other lipid categories, but we'll talk about fats, and then phospholipids, and then steroids. So to remind you what hydrophilic and hydrophobic mean. So hydrophilic means that the substance has an affinity for water because it can hydrogen bond with water. So anything polar with partial positive and partial negative charges, or anything ionic with a complete positive or complete negative charge, those things are going to be able to interact with water, hydrogen bond, and we call them hydrophilic. Hydrophobic substances are the ones with the nonpolar bonds. So lots of carbon-hydrogen bonds, those are going to be nonpolar, which means those give the molecule a hydrophobic character. So they're not going to hydrogen bond with water. They don't get repelled by water, but they just don't. Water prefers to be hydrogen bonded to itself. It's more energetically stable that way than mixing with the nonpolar molecules, which are not going to hydrogen bond with them. So oil and vinegar is the classic example. The oil is hydrophobic, lots of carbons and hydrogens. It's a lipid, it's a fat. And the water is, of course, hydrophilic to itself. So you can see here's a hydrophilic protein. You can see the hydrogen bonds happening between the partial charges. And we also have hydrophilic ions. So they can also partially, they can bond to the partial charges with hydrogen bonds as well. So when we get to these hydrophobic fats, you'll notice lots of carbons and hydrogens, big long tails in this case. That's what makes it hydrophobic. This is the basic structure of a fat. You have one glycerol molecule. That's an alcohol, the OH groups. So it's the small one that you're going to build on. So for a fat, you'll put these fatty acids onto the glycerol. Of course, this happens by dehydration synthesis, removal of water to create a covalent bond, like almost all the building does for macromolecules. And in lipids, or they're also known as triglycerides, you're going to actually add three tails. So a lipid is a three-tailed structure. All three of those tails have lots of carbons and hydrogens. And so this is what we call a fat molecule. Fats are important in a large part for energy storage. So they are very compact molecules compared to carbohydrates. So animals, organisms that are moving around, they frequently store their lipids as, store their energy as lipids because that's going to be portable. Plants don't move around from place to place, so they use starches and carbohydrates to store their extra energy because they can be bulky and it doesn't matter that much. There's also fats cushion vital organs, so your kidneys, those are surrounded by fat deposits. If you ever do a dissection of a mammal, you'll see the kidneys kind of stuck to the back wall with lots of fat around them, so it protects them. Adipose tissue is the fat tissue in your body, and it's important to have enough of that to keep you warm and have your energy stores, but not too much, which then leads to problems like heart disease and strokes. And finally, the insulation, that's what kind of just mentioned, is the extra layer of subcutaneous fat, the fat right under your skin. Mammals have that, and that's going to allow them to keep their body temperature higher without having to use so much energy to do that. So in the fat group, there are two types of fats, or three types of fats. Here are two of them: saturated fat, unsaturated fat, and then our trans fats. So saturated fat, the reason it's called saturated is if you looked at these tails, these fatty acid tails, all of the carbons will be full of hydrogen. This is a saturated fat. So you see every carbon has the max amount of hydrogens bonded to it. So this means that the tails are straight. So you can see straight tails. Here's an example. That means that these pack really well together because they're flat or straight tails, which leads to the saturated fats being solids at room temperature like butter and lard, and they're frequently the ones from animals. So these are the ones we have to watch out for eating too much of them. There's some discussion now how detrimental saturated fats are, but data in the past has suggested that more saturated fat leads to or correlates with higher cholesterol levels and heart disease. So unsaturated fat, those are the ones that are liquids, or we call them oils frequently, mostly from plants and fish. And you'll see that their tail is different here. So one of these fatty acid tails is kinked, and that's because of this double bond. So the double bond makes the tail bend, and actually in this double bond you see these hydrogens are both on the same side of this double bond. So here's the bond between these two, and the hydrogens stick out on the same side. We're going to see that that is different than the trans fats. So this double bond, it can be more than one of the tails too, means that the packing is not going to happen, which means it's liquid at room temperature. These are the ones that you prefer to eat if you're choosing different fats to cook with. Unsaturated fats, a good choice. And then we have trans fats. So here is the unsaturated fat. See how the hydrogens are on the same side of the double bond? Then what we can do is, there's a little bit of trans fat made naturally, but most of it is artificial. And that trans fat, you can notice that the hydrogens are now across from each other. So when you bombard the unsaturated liquid with hydrogens, you shoot them at it basically, sometimes it'll get on the opposite side, and then it straightens the tail out because the opposite sides kind of push the tail up or straight. And now we have a tail that can pack again. So this is going to be things like classic Crisco shortening, a number of margarines, particularly the classic margarine. Those are trans fats. Trans fats are also, if you look at the labels of food, partially hydrogenated vegetable oil, that equals trans fats. So they're partially, they have the double bond, so they're not completely saturated, but they're partially hydrated or hydrogenated. So we're really worried about trans fats. Turns out they're really bad for you, and so you have to be careful to avoid them. Unfortunately, they're in lots of food for texture, and they stay good for a long period of time, but they're required to put that information on the back of the packages now. It's a little tricky though, because if it's under 0.5 grams of trans fat, at least right now, they don't have to report it. So what could happen is if you eat, 4 servings of a food that looks like it has no trans fats, but it really has like 0.4 trans fats, all of a sudden you're up almost to 2 trans fats because you've eaten more than 1 serving. So that can be a little tricky. It will always say though partially hydrogenated vegetable oil, or sometimes some version thereof. So look for that and avoid it. These are the kinds of things that could happen with saturated fats and trans fats. Here's a normal artery, and here you can see a plaque in an artery, so it's clogged there. And some of the kinds of fatty acids and fats that help prevent clogging like that and don't contribute to it are omega-3s. So omega-3 fatty acids have a double bond at the third position from the end, and they're going to be really important for preventing cardiovascular disease. Those are the ones you get a lot in like salmon and a lot of fish, types of fish. Okay, so that's the fat category. Second category of lipids that we're going to talk about are called phospholipids. Not surprisingly, these have a phosphate in them. Same sort of setup with the glycerol molecule here and the tails, fatty acid tails of carbon and hydrogen. But now instead of a third tail, we have a phosphate put on. There's some sort of modifying group. They're different for different types of phospholipids. We're not going to worry about those groups, but they are there. What we are going to worry about is the fact that this part up here with the phosphate and the glycerol, you notice phosphate's negative. So negative means that it's hydrophilic. It's going to hydrogen bond with water. So it would hydrogen bond with the partially positive hydrogen of water. So this part is going to interact with water well. The tails are the same as the fats, where they're carbons and hydrogens. It's nonpolar, so they're not going to interact with water well. This is how we can draw an example or a representation of a phospholipid, and that's what you'll see in a lot of drawings. And what ends up happening is you have this hydrophilic head, two hydrophobic tails, and that ends up orienting itself in aqueous solution in water, which is living system environments, to where the heads face both outside and inside, and the tails are going to be away from water. They sort of hide from water. It's more just energetically more stable for them to be away from water. So this is where you're getting the cell membrane. So this is a lipid, phospholipid bilayer. You see there's two layers. Heads to the outside and inside where the water is, hydrophobic tails on the insides. So those are for cell membranes, making cell membranes. You'll notice these have two tails, phospholipids in biological systems. Some phospholipids only have one tail, and so they behave in a similar way. This is one of the ways soaps can work, is that you have a hydrophilic head, and you have a single hydrophobic tail. In this case, they're like more of a triangle shape, kind of like a cone shape instead of a cylinder. So when you put a bunch of cones together, it makes a ball. And inside that ball, hydrophobic substances like grease and fats, those can be surrounded by the hydrophobic tails, and then the hydrophilic heads touch the water. So all of a sudden you can sweep away the grease, even though it itself won't mix with the water. So you make this little bridge. It's called a micelle. Is what it's called when it has single tails and it forms the ball. And finally, the third one is called steroids. So you've probably all heard of steroids in the context of anabolic steroids, which are also part of this group. But steroids are more diverse than that. So a steroid basic structure is this backbone of four fused rings. Okay? So then you add different types of functional groups onto those rings and you get different types of steroids. Remember, all the carbons are the corners that are not marked, and anything that's not marked with another group is a hydrogen coming off of it. So this would really be a carbon here with two hydrogens coming off of it and then bound to two other carbons on the sides. So here's an example. Cholesterol is an extremely important steroid. You can see it has functional groups and this little group sticking off here. Notice lots of carbons and hydrogens. It's hydrophobic, just like all lipids. In animals, we have cholesterol in our cell membranes. They help regulate the fluidity of the cell membrane, so that's very important. Of course, you don't want too much cholesterol because that can lead to plaque formation and cardiovascular problems. But you do need some. And you need cholesterol too because all other steroids are originally come out as cholesterol and then they get changed to other steroids. So for example, estradiol, a form of estrogen, and testosterone started out as cholesterol in the animal body and then got modified into the estradiol or the testosterone. So both of these are vertebrate secondary sex characteristic hormones. And, estradiol high levels make female secondary sex characteristics, testosterone at high levels make male secondary sex characteristics. And so the fused rings, you'll notice, so these are steroids. So steroids include cholesterol. They include a number of hormones, with estradiol and testosterone as examples. And so overall, they're regulating the body, the hormones are, regulating, coordinating systems, and the cholesterol is in the cell membrane of animals only. Okay, that's all I want to go over for us now. 3 types, what they look like, and what their functions are.